Hello, everyone, and welcome to today's live broadcast. Visualize more biomarkers and extract more insights, moving from single marker IHC to multiplex IF. My name is Christy Jewell, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Ultivue. For more information on our sponsor, please visit ultivue.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Also, please notice, you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of your presentation window, or you can report your problem by clicking on the Ask a Question box. This presentation is educational and thus offers continuing education credit. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's presenters. Scott Lawrence, Associate Scientist, Cancer Genomics Research Laboratory, Division of Cancer Epidemiology and Genetics, NCI-FNLCR, Leidos Biomedical Research, and Dr. Katir Patel, Associate Director, Biomarker Strategy and Applications, Ultivue. For complete biographies on our speakers, visit the Biography tab at the top of your screen. Welcome, gentlemen. You may now begin your presentation.
Thank you for the introduction, Christy, and thanks to everyone for taking the time to attend our webinar today. As we all know, the tumor microenvironment is incredibly complex and is characterized by complex immune phenotypes, dynamic cell-to-cell interactions, and ever-changing tumor evasion mechanisms. Further adding to this complexity is the number of different treatment strategies and therapies that have been developed in the immuno-oncology field. This creates a significant challenge in determining the right treatment for the right patient at the right time. Multiplex IHC is a tremendous tool to help address this specific type of problem. Multiplexing allows for a deep characterization of each unique tumor microenvironment while maintaining spatial context. The biological insights that can be pulled from this information is incredibly powerful. And on that note, I would like to highlight a very simple yet elegant example of this. Here we have a melanoma that's been stained with our InSituPlex technology. What you're seeing is DAPI in blue, granzyme B in that green color you can see there in the lower legend, CD3 in red, and SOX10 melanoma tumor marker in a light blue cyan color. Within this melanoma, we are capturing a biological event by visualizing cell-to-cell interactions in context while maintaining that spatial resolution I mentioned earlier. I'm going to zoom into a very specific region within the center of this image. Here we can see the immune response with a cytotoxic T cell recognizing and preparing to attack an abutting tumor cell. What you're actually visualizing in red is the CD3 positive T cells clustered around a group of SOX10 positive melanoma cells. In the very center, you can see that the membranes of the CD3 positive T cell that has granzyme B polarizing to the membrane, to the lower membrane side, abutting a SOX10 positive tumor cell. What you're seeing is a cytotoxic T cell recognizing and preparing to attack that specific tumor cell. We can clearly see the immune recognition demonstrated by the alignment and flattening of these two membranes. The ability to multiplex the numerous markers, some of which co-express on the same cell, on the same slide allows us to visualize this type of immune response and see this immune recognition and reaction to this particular tumor. That's a simple example of why being able to multiplex within this field of immuno-oncology is incredibly important. There are a variety of multiplexing platforms that are currently available within the market. There are many different considerations to evaluate when trying to decide on the particular type of platform that is best for evaluating your own specific research goals and needs. Ultivue has been really founded on a foundation of evaluating all the platforms that are currently available and picking and choosing the best features of all of them and adapting to what the specific needs of our researchers today in the immuno-oncology field are requiring. Some of the key areas to consider when looking at different platforms is, a high level of multiplexing, be able to specifically target numerous markers and adjust the level of plexing quite easily. We also want to be making sure we can have whole slide imaging. We can see clear and very defined cell morphology. We can visualize cell-to-cell interactions and really characterize any types of immune infiltrations and unique characteristics of the tumor microenvironment. Other considerations where I think many other platforms fail to really establish a wider breadth of usage in the space is in workflow compatibility. You want a workflow that is very simple and can work with many pieces of instrument that you already have within your own laboratories. And then on top of that, really from a data deep dive perspective, you want to be able to identify complex phenotypes. And this requires the ability to visualize co-expressing and co-localizing markers, as well as see things like differential expression of certain types of functional markers such as PD-L1. To highlight some of the technology advantages that Ultivue presents, we're showing a melanoma stained with our PD-1 kit, which has CD3 in red, CD45RO in green, and PD-1 in that light blue cyan color. We've actually dropped off the tumor marker in this case just to allow for a little bit more clarity and visualization. On this slide, I'm really hoping for you to be able to visualize the co-localizations of some of these markers, as well as seeing some of the indications of dynamic range from a qualitative sense. What I'm highlighting here are some single positive CD3 positive T cells. Here we're seeing some dual positive CD3 and CD45RO memory T cells. And please keep in mind when you're visualizing these in any of these analysis type softwares and tools, you can actually get new colors when you stack two colors on top of each other. So when you have red and green, for example, you get this yellow color presenting a co-localization of two markers. We're also seeing exhausted suppressor T cells that are CD3 and PD-1 positive here, and they're in that pink to purple color. And then we're also seeing triple positive cells that are here in this white color. One of the other things I'd also like to highlight as you're kind of visualizing or walking yourself through this image, you'll see cells of different colors and different color variations. You'll see some that are more orange, some that are more pink, some that are more purple. That is very qualitative, kind of just your eye test, showing that there is a significant dynamic range within the staining of our assay here. That you can see low expressors to medium expressors and high expressors. And now if you were to plug this into any sort of standard analysis tool, you'd really be able to further dive into that and really start to threshold and categorize these different ranges of expression into new unique cell populations. One of the other pieces I'd also like to highlight was workflow that I had mentioned earlier. So the UltiMapper and Ultivue technology can be run automated on the BondRx or manually, and it's a very simple, fast assay, about 5.5 hours. Whole slide imaging, it can be done on any whole slide imaging platform. It does not require any types of spectral mixing, so your basic whole slide scanners are fully adaptable to this platform. And image analysis is completely user dependent. This is really showing the fact that we can have a very agnostic approach to being able to deploy our assay to a variety of research laboratories with a variety of different configurations as far as workflow goes. What we present here is a standard workflow that's very amenable to standard histopathological workflows that we see in the clinic today. So now to give you an overview of the UltiMapper assay itself, it is actually quite simple. Ultivue uses antibodies that are conjugated to unique single-stranded DNA barcodes that you can see there on the left. These unique DNA barcodes are unique to each individual antibody. So each antibody has its own unique DNA signature, just like people do. So CD4 would have its own signature, CD3 would have its own signature, CD8 would have its own signature. This allows us to eliminate the need to use any sort of secondary antibody, which helps eliminate things like nonspecific and background staining. This also allows us to use any species of antibodies we see fit. They could be all of mouse or rabbit monoclonals, does not matter. Species is completely independent. The assay itself also allows us to do a single cocktail antibody staining step. and also only utilizes a single antigen retrieval step. There are no rounds of stripping antibodies off and restaining. This is a single primary antibody staining step that immediately follows a single antigen retrieval step. Once our primary antibodies have been bound, we actually go through an amplification step where we amplify the DNA barcodes via tandem repeats. This amplification is just an amplification of that single-stranded DNA. So it's a single-stranded DNA that will be amplified. And this amplification is linear and occurs at the same rate and at the same time for all targets that are there. This is actually quite interesting, especially when we're discussing things like dynamic range. This linear amplification allows us to see a wide variety of expression of certain functional markers such as PD-L1. It allows us to differentiate low to medium to high expressors because of this linear amplification. In comparison, when you're using something like an HRP conjugate, that type of amplification is actually generally much more geometric, meaning it tends to hit saturation quite quickly. And that means your medium to high or medium expressors tend to hit saturation quite easily, and it's harder to decipher or separate the range of expression between those types of cells. Now, getting back to what we have here, we also have a single detection step. And our detection step is also quite simple. It's a simple DNA hybridization where we use complementary strands called imaging strands that have fluorophores conjugated to each one of them that will go in and hybridize to their specific DNA complementary strand or DNA barcode that we were talking about earlier. We simply use a FITC, a TRITC, a Cy5, Cy7 type of fluorophore scheme, meaning we don't need to do any spectral mixing because these fluorophores are very well isolated and do not have any sort of spectral overlap or bleed through. And this allows us to go ahead and visualize the targets we have laid down at the initial step. And also another thing to consider with this assay is that since we're not using secondaries, we're not providing any sort of we're not providing a technology that causes any sort of covalent bounding or caking of things like such as a tyramide platform may do. It allows us to quite easily co-localize numerous markers within even the same cellular compartment at the same time. And this technology also allows for the tissue to be maintaining very, very good tissue quality and integrity, meaning that the assay can be used for other downstream types of analyses. We've had a number of customers that have actually started stacking things like ISH and FISH and things like that after running our fluorescence assay. As you can imagine, only having a single round of antigen retrieval keeps that tissue quite well intact. One of the simpler things that we do on a regular basis is we run H&Es of the same slide. Right? So many workflows that people have seen working with fluorescence, they'll typically have a serial section that is stained in H&E that is generally very, very good for the pathologist to go back and review. Pathologists are trained in bright field. They're trained in pattern recognition, and sometimes fluorescence can be a quite different platform to interpret and read. So being able to get an H&E of the exact same slide you're doing your fluorescence assay on has been a tremendous asset to our pathologist friends out there that are adopting or working with fluorescent-based assays. And to kind of highlight that, I want to show is a very nice registration of the fluorescence overlaid an H&E. And there's nothing unique about this protocol. So we run our standard fluorescent assay, and following that, we will actually just decouple to the side and run a standard H&E protocol to be able to get this functionality and visualization done. So Ultivue's current portfolio has been designed around really focusing on the major areas of research in immuno-oncology today. That's allowed us to derive a number of different kits that are specifically designed to help answer specific questions that are generally faced by most immuno-oncologists in the market as we see today. But that's something that we've quickly learned is not enough for what people are currently doing. We have a services business that currently runs up to 8-plex assays, and it's expanded its capabilities to a fixed 12-plex assay. Our services business can provide staining, imaging, as well as analysis. And so those are all things you can feel free to reach out to us afterwards to discuss. But what we quickly found out is that running standard 4-plex assays in the field is leaving customers wanting more. Based upon the demands that we're seeing from the field and our customers, and specifically with some of the help from Scott Lawrence and his team over at Leidos, we've been able to start to deploy a higher plex workflow to the field. This, like I had mentioned before, it was standardly a services offering for Ultivue, but now we're expanding this higher plex workflow to the field, allowing customers to run 8 plex panels in their own laboratories. And so quickly, and Scott will touch upon this later in his presentation, I'd like to walk you through how we actually achieve an 8-plex without the need to do things like spectral mixing or stripping or restaining of antibodies. So to start, we still start with a single DWAX and antigen retrieval step. We can still apply a single cocktail of primary antibodies, and in this case, we're actually going to apply 8 antibodies at the same time versus 4. We will also still follow that up with a single amplification step where we amplify all 8 targets at the same rate, same time, and again, that's a linear amplification. But when we come to visualization, this becomes a 2-step process. In the first round, we will actually go ahead and image 4 of the 8 markers that are down on the tissue, just in the same method that we had described before, using complementary imaging strands with fluorophores conjugated to them. We will acquire that image on the fluorescence scanner, and then we will run through a process called exchange, where we do a mild signal removal which dehybridizes those imaging strands and removes all that fluorescent signal. This leaves all those amplified DNA barcodes intact, even the ones we just visualized. What we will do next is we'll actually come in with a second set of imaging strands, but these imaging strands are specific to the second set of markers we did not visualize in the first round, so the second set of 4. We are still using the same standard FITC, TRITC, Cy5, Cy7 fluorophores, but this time they're specific to a different set of antibodies. Once we have those down, we will actually do another round of imaging, and then we will co-register the 2 images and fuse them into a single image where we can begin to work our analysis. So you will have an 8-plex, 8-marker assay on a single slide. And with that, I'd like to hand it over to Scott Lawrence, and he will be touching on this as well as a lot of the great work him and his team have been undertaking over the last year and a half since we've been working with them. Thank you.
Okay, great. And hello, and thanks, Katir, for the great overview of Ultivue technology. Today, I'll be discussing how my lab utilizes 3 assays for evaluating protein expression on histological slides. As this technology has grown from single-plex IHC to the current state of running 30-plus markers on a single slide, the scope, fidelity, and resolution of data that can be extracted has grown. We'll be taking a look at these technologies and contrast them, taking a quick look at the strengths and weaknesses of each. But first, we have the requisite disclaimer slide to go over. I'll just show that for a little bit. So, with my group, at MDPL, we are a full-service histology lab that primarily supports DCEG researchers, which is the Division of Epidemiology and Genetics as part of the NCI, with their pathology-based studies. It was decided early on to build the group to be fully digitally integrated from the wet lab through pathology and data analysis. For chromogenic analysis, we provide routine H&E staining for manual pathology assessment. Also, we support singleplex and duplex IHC for marker analysis. We are excited to be working with Ultivue, bringing multiplex immunofluorescence this year to the lab. While these technologies have their role in the lab, in general terms, as we move from H&E to multiplex IF, we're able to get more data from each slide, but often sample quality needs to be higher and lower slide throughput should be expected. Image analysis in our lab is supported by Halo, along with R and Tableau for data analysis. While the trend for data density and complexity moves towards multiplex IF and beyond, advances in deep learning and AI analysis promises to yield more data from all these technologies and is something we're exploring in future technologies. Even with multiplex IF assays capable of generating data from 8+ markers from a single slide, there's still a role for singleplex IHC. Simply, singleplex IHC is fast and robust. Here, we've optimized our PD-L1 assay across multiple clones and run concentrations. looking at the optical density and positive counts using our calibrator slide. The analysis is straightforward and clearly one can see 2 clones failed and move us closer to solidifying our run conditions. To the right is a typical output from our analysis software and some of the metrics we generate when we run a singleplex IHC. While singleplex IHC excels for analyzing a standalone marker, it isn't necessarily the best technology to use when multiple markers need to be considered. Here, we provide some archival images that we are hopeful to run spatial analysis across the markers. But once we took a quick look, we realized that this set would not be suitable for multi-marker spatial analysis studies. A quick glance at the image series notes that multiple issues from incomplete sections, section vibration artifact, and inconsistent background staining. Alone, these issues present their own challenges and are further compounded when trying to get associations between markers. When considering running a multi-marker analysis with singleplex IHC, the experiment should be implemented with this expectation. But even with the best expectation, there is a risk that subsequent sections will be compromised and may not be suitable for registration and spatial analysis. Consider minimizing the number of sections needed, which will limit the amount of markers that can be compared. It is also noteworthy how marker expression can change from section to section. In this data, we wanted to see how the masked regions of CK positive, denoted by red staining, and how the marker expression within, here PDL1 with DAB, would change the more distal we went from the origin section. At section one section apart rather, we do not see a large difference in marker expression, roughly 4%, but by the time the sections are 20 apart, around 100 microns in total, the tissue composition and alignment have largely shifted from the origin section, and we see an increase in marker area difference. It might be hard to see, but note that the loss of the stromal tract from the annotated boundary when sections are 20 apart. Large changes in morphology can certainly alter biomarker expression when doing these focus region studies with singleplex chromogenic IHC. Needle core biopsies have special considerations when considering multiple slides, as they tend to be thinner and with less representative tissue content. We looked at a number of needle biopsies to see if we could correlate nucleic acid yields to tissue density. From these studies, we looked at how the tissue area changes at 10 and 20 sections away from the origin section. At 10 sections away, approximately 26% of the biopsies had an area difference of greater than 20%. By the time we were 20 sections away, in some cases more, 64% of the core biopsies had a tissue area change of greater than 20%. The large changes in frequency and tissue area observed in the core biopsies was attributed to the initial biopsy size and how the biopsy was oriented, whether flat or tilted, or whether fragmented or intact. Due to the nature of needle biopsies, it is less likely that these would be suitable for multi-marker singleplex IHC studies that require spatial analysis. For this, we moved to running a duplex IHC. For the next study presented, we explicitly sought to run a serial spatial analysis across 8 markers, 6 of which are shown here. Each analytical marker was paired with a pan-CK as a duplex IHC. After scanning, the images were aligned and then registered. With analysis, we created 2 data sets. The top one, which has pan-CK on each side, we were able to get each analytical marker's relationship with cytokeratin, including within, outside, and proximal to. On the bottom one, we also generated a spatial map for all the analytical markers to observe relationships between the cell subpopulations. The primary advantage over singleplex IHC was to confidently analyze for our key metric the marker relationships to cytokeratin, with less concern in changes to tissue morphology or drop sections. As an added benefit, we were able to get some interesting data on the spatial orientation between these markers. Ultimately, however, a new population of cells gets sampled at each section, preventing multi-marker single-cell phenotyping, as we'll get into in the next slide. Here is the same registered image stack up close. While the sections are fairly close, they are not perfect. At close inspection, we can see a different population of cells being sampled with each section. Though it appears at least two sections have the same cell being representative, by far most of them are not being sampled twice. Having the phenotyping marker pan-CK here in a Duplex IHC is helpful for doing targeted region analysis, but for looking at single-cell phenotyping, we need a different technology, and that's where multiplex IF comes in. The multiplex IF has a number of advantages over the prior two technologies. Key to this technology, at least for my group, are the tissue conservation and single-cell phenotyping with multiple markers. Also, preserving sample integrity when considering orthogonal analysis such as downstream genomics is very useful. But multiplex IF is not without concerns. Loss of morphological content compared with chromogenic assays can be an issue. This can be mitigated by registering an H&E slide, a multiplex IF image, or just having one of them as an adjacent section. Slide throughput can be a concern if the lab runs 100 to thousands of slides weekly. Multiplex IF prompts thoughtful experimental design. Due to the cost and the previously mentioned throughput, it is not often practical to run every slide possible and filter the data later. Though cost issues are debatable, since less slides are being used overall. Another con not listed here is the increase in image file sizes, needing larger data storage. Large files can take time to move, and storage can be limiting. Advances in storage technology and/or leveraging cloud storage can be a solution for some groups. So here we ran a pilot study with Ultivue roughly a year ago using their UltiMapper IO PDL1 kit. For this kit, it stains for PDL1, CD68, CD8, and pan-CK, along with DAPI for the nuclei. Using our analysis software, we analyzed for marker spatial relationships to cytokeratin, as the top figure shows, and we're able to phenotype each cell and count the population of phenotypes represented in each core. Expanding this analysis to the entire TMA and associating available metadata, we developed a high-level understanding of multi-tumor response with these critical markers. We plan to expand testing to TMA specific to breast and lung cancer to get a broader understanding and assess assay performance on these cancers specifically. Of course, the TMA does not fully depict the heterogeneity we'll see in a test population, but the quality of the staining and analysis possibilities prompted us to move forward with Ultivue and putting together an Aplex panel to run in production. But before I get to the Aplex panel and a preview of what that looks like, I'd like to contrast conventional multiplex IF with Ultivue IF, as traditionally getting into multiplex IF was challenging and cumbersome to many groups. So conventional IF can take months to get to production. Shown is an example of a fourplex arrangement to assess for background staining and signal crosstalk. Panels like this can be rearranged multiple times during optimization, and it can often be challenging to find the optimum fluorophore to target and to balance marker expression. Here is an example of how all fluorophores are not created equal, with exposure held constant, and both Ki-67 primary antibodies and secondary antibody conjugates run at the same concentration Chi-67 detected with Alexa 594 clearly has a higher signal-to-noise ratio over Alexa 405. Additional signal amplification further improves signal-to-noise. The amplified gamma H2X plus Psi5 run at 1/10 of the non-amplified concentration outperforms, as noted by the linear intensity profiles. A clear strength, and one of the main reasons we appreciate Ultivue's assay, is the approach to signal enhancement through linear amplification. This is critical for my group as we deal with archival samples which can be unpredictable at best and often have issues with autofluorescence. This is a plot of autofluorescence for multiple tissues with multiple fixatives and storage ages from source blocks. While the trend is the same, starting high in the 400 nanometer range and diminishing towards the 800 nanometers, the amplitude varies greatly. A well-sorted amplification strategy can mitigate some of this by exposing out the AF, the autofluorescence. Here, the beta-catenin in orange is amplified, and the other fluorophores were detected without amplification. Since the capture was quick enough in the beta-catenin channel, the underlying red blood cells autofluorescence was not captured and would not pose an issue during analysis. On the last slide, I present a simplified model of the three assays discussed today and the corresponding analysis output we generate from each. First, yes, the manual annotations could address limitations with singleplex IHC, but for this purpose, we are assuming automated detection in targeted regions. With singleplex IHC, we can expect getting the number of cells that are marker positive and some spatial data between the cells, such as analyzing for cell clusters. For duplex IHC, we include all the data from the singleplex IHC, and now we have a phenotyping marker like PanCK that lets us separate regions and analyze content plus or minus the phenotyping marker and proximity to. Multiplex IF allows us to do all of the prior, but now adding the multi-marker cell phenotyping and the spatial relationships between those cells and one or more phenotyping markers. In the end, all these assays have their place in the molecular pathology lab. It comes down to a question of balancing the analytical need to the lab's infrastructure and support, samples, throughput, and ultimately cost. As an early preview, here's an example of an image from our aplex multiplex IF we worked with Ultivue to help develop. The markers include CD3, CD4, CD8, CD68, OXP3, PD1, PDL1, and pancytokeratin. We plan to use this panel to assess the tumor microenvironment on breast and lung cancer samples. As shown here, we will be able to pull a number of distinct cell phenotypes, including proximity data associated with each. The high resolution and fidelity of the marker data is critical to our success. All of this will be run in our production environment, and we are very much excited to be partnering with Ultivue and look forward to collaborating more in the future. With that, I would like to thank all these folks that made this possible. Thank you for your time, and now we'll open up to live questions.
Thank you, Scott. And thank you, Katir, both for your informative presentations. Now, as Scott mentioned, we will now start the live Q&A portion of our webinar. To our audience, if you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have so many great questions already coming in. Let's start with this one. Gentlemen, can you talk more on the process to swap one or more markers to make a custom 8-plex, such as novel targets, clone selection, time to deliver?
Sure. So this is Katir. Thank you, Christy, for that question, and thank you to everyone that attended the webinar today. So with that, Ultivue can actually swap in as many markers that are novel or new that we need to. So even if it's a proprietary target or a marker we haven't worked with in the past, we really have to just do a monoplex optimization, and then we can include it into our assay with any of the markers that we've already kind of presented here today.
Thank you, Katir. Our next question. With this technology, can you detect surface and intracellular markers at the same time?
Yes, we absolutely can. I think if you take a look at the end of Scott's presentation, you can actually see a couple co-localizations of multiple markers in different cellular compartments, such as looking at, Tregs, for example. We have CD3, CD4 as surface markers, but you're also detecting FOXP3 as a nuclear marker as well.
Yeah, and if this is Scott, if I could just tag a little bit on that. In our experience, we don't really have too much of an issue between looking at the different spaces, particularly with FFPE sections. We've had some concern when we do fresh cells and looking at cytopreps, but in our experience with FFPE, there hasn't been an issue detecting them in multiple locations.
Yeah, and I think many of the challenges most people typically face are usually trying to detect multiple markers within the same cellular compartment. So, maybe trying to look at one to 2 to 3 markers rather within the nucleus, or 2 to 3 markers on the cell surface. Some other technologies typically run into steric hindrance issues, but with our technology, we do not really see that problem.
Thank you. Can you have artifacts and background signals sometimes? And if so, how do you deal with it?
So a quick point I can make on that is, with fluorescent IHC and many regular standard IHC protocols, background issues are always something that pop up. With our assay, we don't actually use any secondary antibodies. So the amount of nonspecific and background signal that you typically see with standard primary, secondary antibody type stains, we eliminate quite a bit of that. And also by using fluorescence, you have a higher signal to background ratio, meaning that even if you do have very slight or faint artifacts, that signal is generally so low it's not being picked up by any analysis algorithm, and it's something that you really visually can't even pick up on compared to the bright intensity of your positive signal.
Yeah, and I concur. With Katir, I think if we looked at that one slide that I had with the representation for the autofluorescence, what we see is that with these amplification technologies, the signal can generally overcome a lot of autofluorescence that's present in the sample, and you're going to still have issues with fold and all those kind of things with artifacts of sample handling and sectioning. It's kind of why we always very much suggest good wet lab procedures to generate the best quality section samples and images ultimately in the end for the data analysis. But the technology definitely enhances the artifact mitigation that we would see in a traditional conventional multiplex IF.
How many slides can I stain and image in a workday?
So I'll comment for what we can do internally, through our services lab and what they've typically done with our 4-plex assay. They can easily get up to 60 slides done in a day. Depending on what your shifts look like, you could even push that number upwards to close to 90. For the higher plex workflow, you can definitely get 30 slides completed in a day as well. But Scott, feel free to comment on what your experience has been in your specific laboratory.
Yeah, so when we fully expect to launch the 8-plex this fall, it looks like we're going to be processing in production about 45 slides a day at full production, and that's including staining, imaging, registering, and analysis. Our project as it stands right now is about 600 to 800 slides, so we'll definitely be leveraging that. That's on one autostainer. We use the Lycobond Max. I think we're getting a second one onboarded as well, so that might also improve our throughput. The imager we use is the Zeiss Axioscan V1, and that certainly has capacity to support 2 autostainers. So it's potential we can get up to 90-ish slides a day, but that's really pushing it. So I think we were comfortable at 45, and I think that's what we'll likely target, the 8-plex.
Now, Scott Katir, can I mix and match markers across the kits?
So as of right now, the kits are fixed, but you can see that we're kind of coming out with this newer 8-plex workflow that's going to be, formally released in the next coming months. But I'd stay tuned until about mid to early June, where you'll start to hear some more offerings where those types of options will become a little bit more readily available from Ultivue.
Katir, are you considering 12 or more plex to be possible and routine in the future with Ultivue technology?
We're always considering the higher number of plex. That's always a common question we get from the field. Right now, we're really focusing on making the 12 plex as robust and efficient as possible. It's just as we have done with the 8-plex, and we've got the 8-plex to a place where it's readily deployable to the field now. The 12 plex is being held within our services laboratory currently, but we're working on that technology to make it more robust to be able to be field deployable. But it's hard to say whether what the next wave is going to look like, but we're always considering being able to increase our plex.
Thank you. Now, is there a problem with eosin autofluorescence interference when multiplexing on an HE slide?
Great question, and I think maybe I wasn't as clear as I should have been for that part. But when we actually run the eosin, the H&E of a multiplex fluorescence slide, we basically run our multiplex fluorescent assay first, capture that image. Once you're satisfied with that image and that image quality, then we would run the eosin, the H&E, afterwards, and then acquire your bright field image. Once you've actually stained with eosin and hematoxylin, you really won't be able to go back to your fluorescent aspect of the tissue and re-image it. So it's really important that you capture your fluorescent image the way you want it before you actually start the H&E process.
Thank you, Katir. Now, does the number of markers used on a slide affect downstream data analysis? For example, is it better to do a few markers on many slides or highly multiplexed in one?
So I can address this one right off. So it really kind of depends on what your questions are and what your experimental design and your hypothesis is. If you just need to get density of, say, CD3 cells, it really doesn't make a whole lot of sense to throw in several markers, tens of markers, onto a slide. That just makes the process more cumbersome, labor-intensive, costly potentially, and you're really not getting an advantage. I'd say where you want to run multiple markers in a high multiplex situation is when you have specific questions on these relationships between the markers you're looking for. Do you really need to look at spatial relationships, say PD-L1 tumor cells and the tumor microenvironment, CD8 cells, etc., adjacent to? Are you looking at relationships between CD31 stained blood vessels and other targets? That's when you start wanting to stack the data on each other. And also, if you're trying to pick a certain cell phenotype, as we take a look in our 8-plex, if I can get this here, and you want to look at these certain phenotypes, then you're going to need to do the kind of high-plex single slide registration with all these markers. For instance, we're looking at exhausted T cells, for instance, have this phenotype here, which is CD3, PD-L1, or PD-1s rather positive. So that's kind of when you want to put the effort in to doing this large data analysis. Otherwise, I would say that if you're just looking at T cell population, then you might be better off with a singleplex IHC.
Thank you, Scott. Our next question. Thank you very much, gentlemen, for this great presentation. How can we avoid cross-reactions between the same source animal isolated monoclonal antibodies? Now, different antibodies might have different affinities, so should we perform different antigen retrieval protocols at the same time for multi-staining?
So I can take that question. So we don't use secondary antibodies. We actually use the DNA barcodes, amplify those, and then come in with complementary imaging strands conjugated to fluorophores to actually detect those primary antibodies that are laid down. And since we're not using secondaries and we're using those barcode conjugates to do the detection, we can have any species of antibodies in our primary antibody cocktail. They can be sourced from mouse or rabbit. It does not matter. And then what was the second part of that question? I apologize.
No problem. Now, different antibodies might have different affinities, so should we perform different antigen retrieval protocols at the same time for multi-staining?
So, here at Ultivue, we make sure we optimize all our antibodies for a pH 9 antigen retrieval, or ER2 on the bond. And we haven't come across anything yet that has required variant ways of doing antigen retrieval, like a pH 6 or something like that. Most good high-performance antibodies that are on the market today are usually amenable to using a pH 9 antigen retrieval. So we've been able to standardize quite regularly on that.
Very good. Do you have antibody panels useful for mouse tissue?
Great question. That's something we're always looking into. Nothing commercially yet, but through our services laboratory, we could put together mouse-specific panels without issue.
Thank you. How long can we expect the fluorescence to be expressing on the slides and detectable?
So that's another great question. Our fluorophores are incredibly stable. If you're using a good, fast whole slide scanner that has minimal, that minimizes photo bleaching and light exposure to the slide itself, we can get repeat scans of, up to 100 acquisitions over a 6 to 12 month period without any problem. General storage is usually 4 degrees in the dark, but I can say from my own experience, I've had slides that I've had in my own bag for things like demos for well over 6 months just in the dark, and they still perform just fine with very minimal changes in exposure time.
Thank you, Katir. Now, do you have field scientists to help show how to do this and help set up the system?
Absolutely. I think it's a great offering that we have here at Ultivue, that we always send out a field application scientist for demos, trainings, and even implementations. We really want customers to be successful with our reagents. I think it's something that the field has generally struggled with, that sometimes, people are left to their own devices without a lot of specific guidance when working with complex reagents and complex assays. So, having someone there that's well trained in that space to walk you through it and, check out the workflow and infrastructure that you have on site, and to basically be able to help consult you with these projects and assays is something we absolutely offer, and we take a lot of pride in doing that and, interacting with our customers face to face.
Thank you, Katir. Will multispectral imaging and unmixing enhance the results, or is it unnecessary?
So I could just talk from our experience. We did take a really quick look, or a close look at multispectral imaging and, of course, technology. And there's a lot of good things about multispectral imaging, but some of the challenge with it is very sensitive to the the expression of each marker and its proximity to each other. So one marker can easily overbalance another marker, and it makes it tough to balance. So there's a lot of optimization that needs to be done. And I'm sure Katir will talk a little bit more about this, but with the Ultivue technology, each channel is spectrally separated sufficiently where you don't see the spectral overlap. So unmixing is generally unnecessary.
Yeah, that's a great point, Scott. And just to add on to what you just said there, when you actually have to do spectral unmixing, it's required to balance this fluorescent signal for each one of the channels or each one of your markers, right? So that means in order to efficiently spectrally unmix the image. So when you're turning up or turning down channels to get them within a specific range of exposures, you're artificially kind of adjusting the amount of signal that's truly coming from the tissue or from the proteins that are present on that slide. So that's what makes the optimization quite difficult, and it actually kind of skews maybe what you're observing from a biological perspective, and can, you're basically turning a low expressor into a brighter expressor and a high expressor, and turning that down into like more of a medium expressor, just to be able to unmix the image. And the idea of using spectral unmixing is to eliminate that spectral overlap and bleed through of fluorophores that are very close. We use fluorescent dyes that are very well spectrally isolated from each other, meaning you don't have to worry about that crosstalk or bleed through from channel to channel. And since we're not doing any sort of unmixing, the signal you're seeing from a pixel intensity perspective is representative of what is biologically happening within that tissue.
Thank you both. Can this multiplex IF be applied on cytochemistry?
So I have a lot of experience doing multiplex IF on cytochemistry preps, and I haven't actually tried Ultivue technology on it, so I would be curious on what Katir would say about it. But in my experience with multiplex IF on cytochemistry, it's really highly dependent on how the samples or cells rather are fixed and prepped. As I think we had an earlier question about looking at the different cellular spaces, membrane, cytoplasm, nucleus. Typically with cytochemistry or cell preps, those membranes can be easily disrupted and can diminish membrane signal to get into the nuclei and stain for nuclear signal. So it is a tricky balance, and there's kind of multiple fixation strategies that can be applied to get the best of both worlds. But I can't speak specifically on the Ultivue InSituPlex for cytochemistry itself.
Yeah, so from my perspective, we actually haven't tested that yet. It's something that we've discussed internally, and it may be something that we evaluate at a later date. But right now, we don't have any data to say how successful we were or were not at staining those types of samples.
Thank you. Now we have time for a few more questions, but I do want to remind our audience that those questions that we are unable to answer live today and those that do come in during the on-demand period, they will be answered by our speakers via the email address you provided at the time of registration. We do have quite a few questions that have come in, and we for sure will not be able to get to them today, but they will be answered. So let's continue. Can the UltiMapper assay be used on fresh sections that have not been embedded in paraffin?
So we don't test that internally formally, but we have some customers that are evaluating that right now as we speak. And it kind of speaks to the same point that Scott had just made. They will require some fixation processing, and that, working that out and optimizing that is what would be required to see if our assay would work optimally there or not. Another consideration to take there too is that most standard IHC antibodies are derived against epitopes that have undergone specific types of fixation. So sometimes secondary sets of antibodies need to be used in order to optimally stain epitopes within fresh frozen, meaning an antibody that works really well in FFPE may not work as well in fresh frozen.
Is there a trial and error required to eliminate background?
From our optimization perspective, not really. We really optimize a primary antibody first, and then we really just need to optimize the appropriate amount of imaging strands. But that's pretty turnkey at this point for the amount of development we've done on the numerous markers that we have.
Yeah, and just to background Katir, that our experience with the Ultivue InSituPlex versus conventional IF, a lot of that's been taken care of, which is really a large weight off of the lab trying to, as I mentioned, optimize on. It's very difficult to get that and exclude background and the noise. The turnkey solution, Ultivue, has been tremendously helpful, and I think that's a very strong advantage of the technology in the linear application, which reduces a lot of that issue.
Yeah, maybe Scott, you could comment on how much time it typically took your lab to develop a standard, 4-plex assay or something using some other type of maybe chromogenic multiplex or other fluorescent platform?
Sure, yeah. I mean, we do a chromogenic. Generally, as I kind of mentioned in my presentation, we don't go above too much because then all those the chromogens can sort of blend and it gets more difficult to deconvolute the signal from one another. But with fluorescence, of course, the issue with fluorescence is not just optimizing the antibody concentration. You also have possibly the secondary concentration unless you directly conjugate it, but then you also have very low signal. So it's really a balance of signal to and the affinity and avidity of the target that you're looking at. But then you also have to balance it against which channel that works best in relative to your antibody. And then you have to look at according to your on-prem equipment to make sure it balances out and spec that out properly. That process just to get set up is pretty time-consuming, but I think in our experience, we were routinely 6 or 8 months to get a 4-plex panel IF into production, and it was a significant amount of effort on our team.
Thank you, gentlemen. And we'll wrap with this final question. What kind of expertise is needed to use your technology?
So I think that's one of the beauties of our technology is that, and it kind of spins off of what Scott was just referring to, is that we really pre-optimize these assays for you, meaning the development and optimization of each individual marker and then the multiplex afterwards is something that you do not need to do. And that in the past has required a lot of expertise and a lot of time, as Scott had mentioned, in order to do so. So, being able to deliver pre-optimized kits is something we can do fast and quick. And assuming the site has, the proper infrastructure to do the staining and the scanning, we can really get anybody up and trained on how to run this assay.
Thank you both for this excellent Q&A session. Do either of you have any final comments for our audience?
Sure. I just want to say thank you to everybody for taking the time to sit on this webinar today. And I want to say a big thank you to Scott and the Leidos team. As I mentioned before, they were really the impetus for us to develop this new 8-plex workflow for the field. As I said before, it's something that we had just kept as an internal technology with our services business, but Scott's been very much a pioneer in the space. And him and his team have been an absolute pleasure to work with and to help us, get us to the next level as well. So thank you all, and thank you, Scott.
Yep. Thank you, Katir, and thank you, everyone else, for taking time out and listening to our talk. And stay safe, everyone.
Thank you both. And before we go, I do want to thank our audience for joining us today and for their interesting questions. Again, we had quite a few questions we were unable to answer, so those questions and additionally the questions that are submitted during the on-demand period, they will be addressed by our speakers via the email address that you provided during the time of registration. I'd like to once again thank Scott Lawrence and Dr. Katir Patel for their time today and for their important research. We'd also like to thank LabRoots and our sponsor, Ultivue, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you today via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Have a great day.